inside the science of designing new life forms. It started with a creation that did not evolve. It was assembled piece by piece, line by line, and once it began to function, biology crossed a point of no return. To understand why this matters now, we need to look at how life was once shaped. Evolution worked through accident and pressure. Mutations appeared randomly. Environment selected what survived. Humans could influence this process, but only indirectly, breed selectively, alter habitats. Life itself wrote the rules. That boundary dissolved when biology became readable. DNA was decoded, cellular processes mapped, metabolism reduced to pathways and logic. Life stopped being mysterious and started becoming legible. And once something is legible, it can be edited. At first, scientists modified what already existed. Genes were added, removed, or silenced. Organisms were improved incrementally, but a more radical idea followed. What if life did not need to start from nature at all? What if it could be designed from scratch? Researchers began assembling genomes synthetically. Custom DNA sequences were written digitally, then inserted into empty cellular shells. When the cell activated, it followed instructions no organism had ever carried before. It lived, it replicated, it obeyed design. This was not cloning, it was authorship, life defined by intention rather than inheritance. Early creations were simple. Bacteria engineered to produce fuel, medicine, or materials, but the method scaled, complexity increased, behaviors emerged that were not explicitly planned, and that is where the tension began. Design life did not remain static. It adapted, it evolved, it interacted with the world in ways no blueprint could fully predict. Once life can be designed, the question is no longer whether we can build it, it is whether we can control what it becomes. As designed life forms moved from theory to reality, control became the central concern. These organisms were built with safeguards, genetic locks, kill switches, dependencies on synthetic nutrients. Layers of protection meant to keep design contained. At first, the safeguards worked. Engineered organisms behaved exactly as intended. They performed tasks efficiently, then stopped. But biology has never respected permanence. Mutations accumulated. Environments introduced pressure. Life did what it always does. It adapted. Some safeguards failed quietly. Dependencies weakened. Control mechanisms degraded over generations. The organisms did not rebel. They optimized. Survival rewarded flexibility, not obedience. This exposed a fundamental contradiction. Design life was meant to be precise, but life thrives on variation. The more adaptable an organism became, the less predictable it was. The more predictable it remained, the less useful it was in dynamic environments. The conflict deepened as applications expanded. Design microbes were released to clean pollution. Synthetic organisms supported agriculture. Living systems became infrastructure. Once deployed at scale, retrieval was impossible. Life released into the world does not come back. Researchers face an uncomfortable reality. Designing life was easier than governing it. Oversight lagged behind innovation. Global coordination proved fragile. One lab's breakthrough could become another region's ecological disruption. The emotional weight peaked when simulations showed long-term outcomes diverging wildly from initial intent. Harmless designs could become dominant species. Useful traits could spread beyond containment. Biology did not recognize human categories like purpose or permission. What frightened scientists was not catastrophe, but irreversibility. Designed life could persist long after creators were gone, shaped by environments no one could anticipate. Once life is authored, evolution does not stop. It simply continues without asking who wrote the first draft. The conflict reached its peak when designed life began interacting with natural ecosystems, not as an isolated experiment, but as a participant. Synthetic organisms exchanged genes. They competed for resources. They found niches no one had planned for. This was the moment control slipped from intention to consequence. Design traits spread beyond their original hosts. Horizontal gene transfer blurred the line between synthetic and natural. Biology absorbed design the way it absorbs everything else. Attempts to halt this integration failed. You cannot recall evolution. Once genetic information enters an ecosystem, it becomes part of its future. Containment strategies assume static environments. Reality was dynamic. The deeper concern was philosophical. Designed life carried goals embedded by humans: efficiency, productivity, optimization. When those values entered natural systems, they reshaped balances built over millions of years. Ecosystems evolve for resilience, not output. Researchers face a paradox. To be useful, designed organisms had to survive. To survive, they had to adapt. To adapt, they had to escape design. Control and success pulled in opposite directions. The emotional peak arrived when scientists realized something irreversible. Humanity had introduced intention into evolution itself. Not selection alone, but direction. Life was no longer only responding to the environment. It was responding to human goals. This changed the meaning of responsibility. Designers were no longer just engineers. They were ancestors of lineages that might outlast civilizations. Designed life did not mark the end of nature. It marked the beginning of a world where nature carries memory of human choice. The resolution did not come from abandoning designed life. It came from redefining restraint. Scientists learned that control could not mean permanence. It had to mean guidance, monitoring, and humility over time. New frameworks emerged. Designed organisms were built to degrade gracefully. Traits were limited in scope. Ecological compatibility became as important as function. The goal shifted from domination to coexistence. This marked a cultural change in science. Creation was no longer celebrated alone. Stewardship followed design. Long-term observation replaced short-term success. Responsibility extended beyond deployment into generational care. Designed life forced humanity to confront a difficult truth. Creation without patience becomes intrusion. Power without accountability becomes legacy, whether intended or not. What science learned in the frontier was not how to build life, but how to live with what it builds. Biology does not freeze at the moment of invention. It continues, learning from the world it inhabits. Designing new life forms did not give humanity control over evolution. It gave humanity a voice within it, and that voice will echo long after the designers are gone. Subscribe to join the journey.